Hi, good morning, everybody, and thank you for joining us today for our briefing on vaccine development and vaccine innovation for the future. This briefing comes at an important time, as we're all aware, as we're seeing new variants and additional mutations of the COVID virus. What we do know is that current vaccines do not protect against all variants, so continuing vaccine development is crucial. This will be addressed later in the presentation. The vaccines currently are effective, but over the medium term can drive new mutations based on a lot of different factors, again, which we'll get into. The vaccine industry has really experienced underinvestment for decades. And before COVID, the last meaningful vaccine was probably about 15 years ago for HPV. With the realization of the importance of vaccines, we need to continue to look at ways to leverage new technologies and approaches to quickly develop vaccines, not only against COVID, but against pathogens that we haven't come in contact with yet. And the past year certainly has shown us the importance of quickly developing broad-acting vaccines. And we're very excited today to have a panel that will share with you the challenges, opportunities, and the innovations that we're seeing in today's vaccine environment. Some technologies that are very important in our opinion are coming out of the Vaccine and Immunotherapy Center at Mass General, where their goal is to really develop and transition promising technologies through preclinical development and into clinical stage development to address new pathogens or to improve the treatment for existing pathogens that might be mutating. 4 of these technologies have recently reached animal preclinical studies or first in human testing, and there currently are 9 vaccines and immunotherapies in development at the VIC. So it's an effort that is really actively involved on a daily basis in research, driving new technologies in vaccines. The panelists are really, we're very fortunate to have them. I've had the opportunity to work with them in various capacities for 5 years now, and that experience has been informative and humbling and has led, in my case, for sure, to great optimism because I see their commitment, this panel, every day to creating better approaches to address new pathogens, new infectious disease, and improve patient care. The panel is unique in that there is experience and perspective that comes from high-level corporate access on the vaccine programs that we all read about, high-level advisory positions to the government, to bench research on vaccine platforms, coupled with clinical experience in the ICU treating patients over the last year. So it's really a unique group of folks. Dr. Michael Callahan is a staff physician at MGH, Mass General Hospital, in internal medicine and infectious disease, and a subject matter expert for international clinical trial design for the Assistant Secretary of Public Health Preparedness at the Department of Health and Human Services. And Dr. Callahan has conducted research in tropical infectious disease, sepsis, and immune therapies since 1986. Interestingly, he previously worked at DARPA, where he ran the $260 million Accelerated Manufacture of Pharmaceuticals program to protect against natural and intentional pandemics and antimicrobial resistance. Mark Poznansky has spent decades working on vaccines and immunotherapies, and he's really an innovator who bridges academic discovery and clinical care. He's worked on a number of new vaccines and immunotherapies for infectious diseases, including COVID-19, as well as cancer and immune-mediated diseases like diabetes. He oversees the research team at the VIC and really focuses on the operations of getting new platform technologies from preclinical programs into the clinical setting and into patients. Finally, we have Dr. Patrick Reeves, who's an immunologist and leader in deep immune profiling. Patrick applies cutting-edge technologies to investigate and visualize immune function with remarkable precision and detail. It's a visual process, which is unique and really adds to our understanding. And he's a group leader in the MGH Vaccine and Immunology Center and an instructor of medicine at Harvard Medical School, and is a recognized expert in virology, immunology, and cell biology. And the goal of his research is really to fine-tune vaccines and therapeutics to meet new and emerging immune insights. So again, that's our esteemed panel. And with that, we will move into our discussion.
Thank you, Patrick. This is Michael Callahan. Good morning, everybody. Part of my responsibilities here is to set up the storyboard and to sort of correct between where we are in our technology development to protect U.S. health security, our people, our military, and our friendly foreign nations where the U.S. is a major contributor. And it would be good to do that against the backdrop of the very recent transition in global high consequence infectious disease outbreaks. It is no surprise to any of the listeners in the Congress, and many of you, we had the chance to brief previously on many of these issues, but the consequence and the frequency of emerging infectious disease involving novel and unprecedented pathogens is really in an uptick, with approximately a 6x doubling in the last 3 decades. Ominously, the type of new agents that we're contending with are increasingly represented by esoteric threats that we could not have contemplated in the past, with the exception, of course, of influenza. And we'll get to that later in our discussion today. Depicted here is a snapshot courtesy of Google Images in several of the other references at the lower right-hand side, including Johns Hopkins, depicting the magnitude by total case mortalities as can be recorded by history over literally here centuries. 2 points are made here. One is that COVID is sitting at number 11, where that red arrow is indicated, with a current case accrual globally of estimated at 2.9 million calculated, and a consequential increase in total mortality, which is much closer to 4 million. And this has to do with the distribution of unrecorded cases among the world's poor, and does not involve mortality from the secondary and tertiary side effects of COVID infection and convalescence syndrome, a critical feature for when we deal with vaccine design. With that in mind, let's turn to the next slide and look at some of the very recent events. And it's important to share with you information that is breaking now. Much of this is for congressional representatives. It is not classified, but it is unknown, and it will be reported in peer-reviewed literatures and by our colleagues at the CDC by Thursday of the coming week. We noted that we are in the middle of the 4th US surge, and this will be the 3rd major surge. It was both anticipated by watching our partner nations in the Southern Hemisphere and watching the inevitability of COVID-19's viral evolution in response to that virus hitting immune people who had seen the Wuhan strain and who were now getting vaccinated, and now the virus was contending with the challenge of bypassing or breaching the immunity that is offered by vaccines from multiple nations: Russia, China, India, the US, and many of our EU partners. So what we're seeing as we speak is a 19% increase in cases over the last 12 days. That means our 80% reduction of cases that were laudable for all the magnificent effort of the last administration in this one in case control and leadership by some really great governors, that 80% decrease in cases is being partially reversed by nearly 1/4 in a 12-day period. This is an ominous indicator for which we have to mount new strategies to contend with both the variant and to work on the concerns about vaccine hesitancy in underserved populations and hard to reach places. Bullet number 2 on the left side is that what we're seeing is that states that have reduced their restrictions, no surprise, or have lower vaccination, again, no surprise, are contending not just with the return of the routine cases, but this is the most fertile ground from which we are seeing the variants, not just the UK variant, but the much more ominous and more deadly P1 variant from Brazil, where they are getting a considerable foothold. So we are in a foot race between the migration of the variant into previously infected individuals who can be reinfected with the variant, and people and populations that are unvaccinated. Our third big takeaway to bring up to leadership is that these problems are compounded not by just the variants we know about, but a large array of variants that number 4,100 COVID-19 strains that have been identified in people. These are not hypothetical, they are not modeled, they are retrieved and sequenced courtesy of US investments in global sequencing and that of our friendly partners. Our takeaway, looking at the right side of that of the chart, is the updated Hopkins trend lines. But what you're going to see on the CDC report on Thursday and Friday is the percentage of those cases that are made up of the new variants, a major subject for developing a resilient vaccine program that is no longer reactionary, but preemptory, that uses strategic vaccine design to produce evolutionary barriers to prevent the escape of the variants of the future. So that we're going to turn to Dr. Hesitancy for an overview of the current vaccine technologies and set the storyboard for how to move from being lucky, which we have been in this nation in the last two outbreaks, to being smart and to repeat the successes of our technological innovation and to learn from our global partners and to be ready to counter the variants and other pathogens of the future. With that, I'll turn it over to Mark. Mark,
you're on mute.
Sorry about that. Yeah, thanks very much for involving me in this session. As Michael said, we are facing sort of an unprecedented time in terms of generating a vaccine fast enough to deal with a pandemic. And what we've achieved, and I'm using we in the general sense, is a rapid vaccine program under the two most significant ones in the field, Pfizer and Moderna, to produce a vaccine that was good enough to halt or at least slow down the pandemic that was occurring across the globe. That required focused production of a vaccine against one specific aspect of the virus, probably its most potent component, which is the spike protein on the surface of the virus. And therefore, under the Warp Speed program, there were 5 vaccines, all with sort of slightly different delivery systems, including the RNA vaccines, that focused the immune response on the spike protein and has shown tremendous efficacy in reducing the most severe aspects of the disease, the death and the severe disease that's associated in hospitalizations. That was the first and critical step. Those vaccines also generated antibodies that were capable of binding to that spike protein and neutralizing the virus. And that was also occurred very rapidly after the vaccine and boost in the case of the Moderna and the Pfizer vaccines, that then controlled both the potential of infection in the individual and also potentially, as we're learning from places where vaccine frequency is very high, like in Israel, are having an impact on the spread of the disease as well. However, these are your first line and recognizably effective ways of stalling a pandemic. But as Michael's alluding to, there's a second generation of vaccines needed that need to be nimble to address more of your immune system, including your T-cell immunity, and also the fact that there are emerging variants that have already escaped from the vaccines that have been produced in the first generation. And it's that nimbleness that we're really trying to address here in this talk, and the need for that nimbleness in vaccine design. Next slide. So in the past, obviously, these are the aspects of vaccine development and the history of vaccine development, using weakened viruses, taking parts of viruses to make an immune response. And then in the context of COVID-19, the essence of this was early identification of a genetic sequence that could then be used to generate the RNA or DNA-based vaccines that then could be used to generate the spike protein in the person who was vaccinated, so that you could evoke an immune response that would then be able to go out and target the virus if you became infected with the virus. And these are the things that have occurred very rapidly under the COVID-19 pandemic. A lot of aspects of the pathway were cleared by regulators. A lot of work was done in parallel rather than in series. A lot of things were done in parallel at risk to the companies. Large amounts of vaccines that hadn't yet been cleared through phase 3 trials were being assembled and built and made, even though the EUA approval had not yet been issued. And then, of course, once it came to the EUA review at the FDA, this was done very rapidly indeed. These were all the adaptations that were made in both the biology, understanding of the biology of the virus, all the way up to the regulatory path that enabled the first vaccine to be approved within 326 days of having identified the genetic sequence of the virus that was being targeted. Next slide. Now, as Michael alluded to, we're facing a new challenge. 4,000 variants of COVID-19, of which have already killed more than 2.7 million people worldwide. We're sort of in a different phase of this, and as that New York Times reporter described it, we have to think of it as almost like a different set of epidemics that's occurring here based on the different qualities of the variants. The most important point being that as soon as you start vaccinating people, the virus comes up against that vaccine and evolves ways of evading the vaccine. And that is essentially what is going on at the moment. And we obviously need to adapt our vaccines to get, in a counterattack, block those escape routes that the virus is taking to evade the current generation of vaccines. And for that, you need great nimbleness and new vaccine platforms in order to achieve that goal. Next slide. So I'm going to hand this over to Patrick here. Patrick Reeves.
Actually, Mark, I think these are still your.
Oh, okay. So I guess what this slide is telling us is that the most important thing here is that we have to be aware that this is not just COVID-19. There could be COVID-21, there could be MERS, there's avian flu and so forth, which are very, very significant threats to the human population. And whereas we would like to feeling of like, well, we've dealt with this pandemic, it's on the wane, it's actually just the beginning of a pandemic era or a pandemic century where we are going to have to systematically address wave after wave of pandemic or epidemic of new emerging infectious diseases. And the consequences of this are, on every single time that we address this, we are going to have to address the safety and efficacy of those vaccines, and also the speed at which one develops immunity as a result of the vaccine. These are all of the challenges here listed that we face in the second half of 2021 onwards. There's not a moment here for resting on our laurels. So we've got warp speed vaccines out there for the COVID-19 that appeared last year at pandemic levels, and we just can go back to the way we were before. There's a significant need for innovation to address this pandemic century that we're facing. Next slide. So the next surge that we'll see, and it's sort of in a way predictable based on where things are currently, is that patients who were infected with the original strains of COVID are very likely to become reinfected this winter. This may well be an annual or biannual type of issue where new variants arise, dominate across the globe or in specific countries, and then we will have to address. The initial spike-only vaccines, a sort of warp speed type model, would have to move extremely fast to be able to adapt to variant selection. And otherwise, we're facing this sort of whack-a-mole problem that as soon as a virus variant arises, we need to be able to get a vaccine out there rapidly to wipe that out, whether it's COVID-19, COVID-21, and the viruses beyond that. I'm just going to hand over to Michael if you want to nuance this particular slide further.
You're muted, Michael.
Michael, you're on mute.
Oh.
I think the admin has to unmute him.
We can move on if that's not working.
Yeah, please go ahead. I'm not sure what the issue is.
Okay, next slide.
I'm back here, if you guys can hear me.
I just wanted you to, Michael, if you want to nuance from the previous slide, you know, this.
Absolutely. The way to think about this as in the congressional outbriefs and the current commission reports reviewing the COVID response is it is clear that we did not invest in Operation Warp Speed, of which I was a member, in 8 separate vaccines. We made one vaccine 8 different ways. We did not make a second and third vaccine to counter the variants, even though our experience with this virus in China, Italy, and our early experience in Europe demonstrated that variants would emerge in immune people who were facing the new viruses, or in people that were vaccinated with a very narrow vaccine that just used spike protein. So the way to think of it briefly is you need multiple multiple targets, like spike and another one, N, which other nations have chosen to invest in to prevent the emergence of variants. So Patrick will talk more about this with the range of targets that you need in a strategic vaccine program. And the whack-a-mole is both demonstrated this issue of how vaccines themselves can work against you by speeding mutant evolution and creating variants, as we're seeing in our foreign nations, which are using similar vaccines and for which are contending with a broader range of variants, some of which are quite devastating, such as the P1 we talked about. So whack-a-mole is not a theory. It's being proven in the clinical experience, and it's one from which our nation is now aggressively working for countermeasures in the very near term. So thanks for that, Mark.
Thank you, Michael. Next slide. So innovations in vaccine development. We were actually funded by the Department of Defense and by DARPA, actually at least a decade ago, to come up with a vaccine platform that could deliver a vaccine and test a new vaccine in less than 120 days. That was the objective, and it talks to the vision that DARPA, Dr. Michael Callahan, had with regards to the needs for a pandemic response vaccine platform years before the current COVID-19 pandemic. There were many great minds, including Michael, thinking about what was needed in the future well before COVID-19 reared its ugly head. The other component of innovation in vaccine development would apply that you can stockpile core parts of the vaccine. So the virus appears, you design a virus targeting component that evokes a T cell response and a B cell response, and you combine it with the stockpiled component of the vaccine that's already there, so that the vaccine itself is 90% ready before any emerging infectious disease is identified. And then once the emerging infectious disease is identified by sequencing and by surveillance, another huge area that Dr. Michael Callahan was a champion of, you then design the targeting part of the vaccine. So they sort of snap together like Lego, ready to prepare the immune system to fight. So the final 10% of the vaccine is added when the emerging infectious disease like COVID-19 or potentially COVID-21, if it appears, is identified. And it basically serves the role of turning the immune system towards responding and fighting to that particular pathogen. And all of this can be tweaked right up to manufacturing. In other words, the targeting piece can be designed really on the fly based on the way that the virus is moving in terms of its mutation, and then nail it as it's emerging into the human population. So there's that component of innovations in vaccine development that were considered necessary actually 10 years ago, and the team at Vaccine and Immunotherapy Center at Mass General, along with other teams in the US, have been developing these types of flexible and nimble platforms to address the needs of rapid vaccine development. Next slide. So handing over to Patrick.
Thank you, Mark and Michael. So picking up where Mark just left off, part of our objective, as he said, was to be able to have a platform that would allow us to move quickly and have pre-positioned components that were static, and then tailored components that address the particular needs of each challenge as it appeared. And so the conception that we've executed on here is, as Mark said, this Lego type approach, where on the left-hand side of the diagram there, you can see that there's this component which serves as the delivery vehicle and as the adjuvant for the vaccine. And this can be generated in large quantities, stockpiled, and is stable for long periods of time. This adjuvant is well described in the literature. We know how it modulates the immune response to then train it to be effective. And then the payload, so to speak, for this platform can be tailored in real time. So we can get the sequence of the virus or the bacteria that's emerging, begin to analyze it, and start the synthesis process. And those targeting units can be engineered to include the necessary components for training two different facets of the immune response. One is the T cells, which are responsible for going in and clearing out the infected cells. And then also, if needed, we can include the B cell training components to make those antibodies. So those targeting units then can be synthesized at scale relatively quickly. And then those two things can be assembled together in a room temperature reaction that doesn't require any specialized equipment. And that assembly is highly specific, highly rapid, and is then ready for delivery. And that stabilized, or that vaccine platform then is stable for long periods of time. It meets, it doesn't have some of the same liabilities as some of the other platforms that it needs rigorous cold chain controls this is something that's stable at 4 degrees and is tolerant of being at higher temperatures. So even a lot of vaccine wouldn't be ruined by a transient movement to a higher temperature. Okay? And this formulation then does not require, because of its structure, doesn't require any other chemicals or adjuvants to help drive the reaction. We don't need to add anything extraneous. These two components alone are sufficient to drive an immune response. So can we move to the next slide, please? So, going more into detail about the process by which this kind of a platform can be generated, as I was saying a moment ago, we can get that sequence information from our partners, or as it becomes publicly available, and then that either DNA sequence or by mass spec sequencing, we can then have the assembled units and start to push it into our analysis tools. And that allows us to collect information from disparate parts of the globe and start to integrate that into the vaccine design. There are a well-worn set of tools then that we can use to then analyze those sequence information. And as part of that, we first predict what components are going to be attractive to the immune system. And then we can refine that list of candidate epitopes and to minimize the number of epitopes that might look something like our own cells, our own proteins, or things that are otherwise beneficial to us, like our commensal bacteria in our intestines. So things that might drive an adverse reaction. We can down select those components. And then there's a further refinement to the actual sequences or the actual components that go into the vaccine to make sure that they aren't going to require highly tailored, specialized chemical processes. We will down select some of those peptides or truncate them so that they don't have some of the liabilities in manufacturing. And then, as I was saying a moment ago, also about the adjuvant component. So this is the left-hand side, the stable unit. This is a highly purified protein that can be synthesized on scale and is well described. We do not have to worry about things like bacterial byproducts or other toxins being incorporated in it. And because of the nature of the vaccine, when it's injected, it doesn't persist for long periods of time, which then can cause a decoy or a depot effect where the immune response is focused, but it's focused in the wrong area. It's focused at the site of injection. And rather, it delivers the payload and then it gets out of the way and it lets the immune system get directly to work. Okay. And then finally, from the very outset, with the vision from the leadership here and from our partners, the platform has been designed for scalability and cost control from the very beginning. That's part of this intentional thing of having a stockpiled core unit and then these very rapidly tailored units, that last 10% that Mark was speaking to a moment ago. And so then you can mitigate some of the costs by having that stockpile component. And then even as you need to adapt your vaccine, it wouldn't necessarily be that all the components of the vaccine have to be remade. As with some of the platforms that are out there right now, as they're dealing with the emerging variants, because it's not tailored, they have to essentially remake the entire vaccine. On our platform, the way that it's developed, it could be that only a subset of the components have to be tailored, and you could essentially hot swap. So as variants start to emerge, you don't have to restart. You could be 80% completed and you have to finish the formulation with those last few tweaks that now give you a more up-to-date platform. Next slide, please. So as Mark was saying a moment ago, we've previously done this in a number of instances where we were able to move from the sequence to demonstrated immunogenicity in a preclinical model in less than 120 days. And I've already talked about the process in general terms of how we identify those epitopes, the payload component, and then we refine it to be more safe for the patients. And the process now is to where we can work with industry partners to understand how we transition this smoothly from academia, from these highly specialized projects, now to something that's going to be robust in terms of manufacturing. And we're also partnered with regulatory consultants and talking with our FDA colleagues to get guidance on how to move this effectively through the regulatory processes. And lastly, as I was pointing out a moment ago, this addresses the very real need for adaptable vaccines that are rigorous in terms of their tolerances for the variabilities in supply chains, rigorous in terms of their ability to be tailored and specialized. And then this doesn't require any of the other things that can elicit concerns, like vectors that are going to have either pre-existing immunity or acquired immunity that limits their usefulness after the first or second shot. Things like other components that are required to drive a strong immune response so that then you have safety and toxicity concerns. So all the way around from the very inception of this project, of this platform rather, we have been mindful of these other constraints. This is not just a purely academic exercise, and that's part of what's so wonderful about the team here, is that with the clinical expertise and the experience of Mark and Michael, along with a lot of our other partners, the whole process has been built in from the very beginning. Next slide, please. So with that, I'll hand it over to Pat, who can talk to you more about how this might benefit from support from you and your seniors.
Thank you, Mark and Michael and Patrick. That's super informative. And just so everybody knows, we are taking Q&A. I meant to mention that at the beginning. So my apologies, but please feel free to submit your questions and we'll have the team answer them. I'll leave it up to the group to read. The company that is moving this platform forward, the technology from Mark and Patrick and Michael from the VIC. We do have two asks in the appropriation process to better fund rapid vaccine development to address variants. And again, I'll leave the next couple slides for you to read on your own, but want to thank the panelists again, and I'll get to Q&A. Just to sum up, we are continuing to see variants and mutations, and the good news is there's people on the front line, as you've heard today, that are focused on trying to, at a minimum, stay in lockstep with what's occurring with mutations, but based on their experience and some technology and some proprietary work that has been done, there's the possibility that we can actually stay ahead of at least some of the variants. So hopefully you got a good understanding of some of the challenges that exist with existing platforms. We believe that there are options moving forward where you don't have to hit reset every time to address new mutations. So with that, I'll stop and we'll go to Q&A. Okay, so there's a question here. Are there other technologies such as contact tracing or even social web tornado warning tech for people that might be applicable here?
I could,
I can take that.
Michael, yeah.
Michael, yeah.
Sure. So first of all, excellent question. To think that just the bricks and mortar technologies of vaccine development that come out of places such as the Mass General Vic or come out of big pharmaceutical companies, that is actually a modest part of the solution. What we need to do is figure out the strategic targeting of vaccines, as well as achieving, to make sure we have the lowest number of non-immune individuals. To your point, there is, for the first time in pandemic response in the US, a robust investment in the use of social networks, in Tornado specifically, and AI algorithms to find not those that will be infected, but rather those that are infectious and propagate the infection. This is the same decision tree that we were in last March of 2020 when we were designing the vaccines. Is we designed vaccines to prevent death and severe illness. Another group was working to design vaccines that prevented contagion. So the point we're making here is there are equal investments that can be done to stop the pandemic rather than to just prevent death and severe illness. So this was phrased beautifully by our European colleagues when they said, let's take those that will die and convert them to the diseased. Let's take those that are diseased and convert them to minimally ill, and so on and so forth. So it's an excellent point, and in our experience, this is the first time non-pharmaceutical measures have been tightly joined with the selection of vaccine design, and it'll be an extraordinary thing to follow in the next year. Thank you for the excellent question.
Thank you, Michael. Another question, and Mark, maybe this is one for you to handle out of the gate. This is the question. When we get to questions about hesitancy to get vaccines because of new mutations and vaccines not covering the mutations, what are the best ways to combat vaccine hesitancy and ensure people get vaccinated with the currently approved vaccines while simultaneously investing in new vaccines?
Yes, so at the top of the list, from my point of view, that I can see is the combination of leadership and sound information dissemination across the nation. And these are things that we're beginning to see now, rather than wishful thinking or opinion-led decision-making. So that's the most important thing at the top level to address vaccine hesitancy, is to inform people that vaccines have been developed that are safe and efficacious, that are reducing the impact of the virus in terms of the causation of death and severe disease, and that in order for those vaccines to be effective and protect not just yourself but others, you need to get vaccinated. The second component of vaccine hesitancy is to identify those within your community and communities that are vaccine hesitant for whatever reasons, and then go after those communities equitably and in a recognition of diverse opinions and diverse cultures that say that a failure to vaccinate any population anywhere in your country is a failure to vaccinate everyone. And that has also been addressed. I've been seeing that very dramatically here in the hospitals in Boston, going after the communities that actually are often at most risk in the cities and towns that have vaccine hesitancy for a variety of different reasons and have not been approached with regards to sound data-driven information. But when that is given, we've been seeing a significant uptick in vaccine adoption by people in those communities. So sort of that top to bottom approach in terms of addressing vaccine hesitancy. For the specific answer, immunity against COVID-19 is likely immunologically to be helpful in terms of your body's ability to raise a response against other COVID-related viruses. There is the potential for cross-reactivity, and consequently getting some protection from the COVID-19 virus is better than no protection from any of the COVID viruses. So that's another important point when, if that aspect of COVID hesitancy is brought up, that why bother protecting against COVID-19? I'm going to get COVID-21. We are pretty sure already that COVID-19 vaccines, as they're currently configured, prevent severe disease and death, which is enormously important. But as time goes on, it may turn out that that immunity that was evoked by the first generation of vaccines is at least helpful in generating immunity against second generation COVID-19, and also the additive effect of additional vaccines.
That's great, Mark. Thank you. Next question, I think, is one for Michael. What are some of the regulatory concerns for rapid vaccine development, and could a large number of rapidly developed vaccines create burdens for national and international regulatory agencies?
Yeah, this is from Ian Banks, and it's a critical question because the regulatory agencies, the actual regulatory agencies that are reviewing the packages for new technologies, the average reviewer, our colleagues, and I was a former worker there, they're now running 91 hours a week. So they are true patriots in terms of delivering a health security advantage and to get through these packages. But specifically to your point, Ian, there's no doubt that the agency is not set up for high throughput and accelerated approval at the pace that is currently needed to counter the emergence of variants. So to this end, the current sitting director, which is Janet Woodcock, is mustering a huge number of resources to expand the reviews to go after the so-called big wins. And what they're seeing and investing in is not the regulatory package in the moment, but the one that's going to get us out of this debacle and convert the pandemic to an endemic, to a nuisance seasonal disease like influenza. The way that's being done is to strategically pick packages from companies and from academic labs and consortiums, including federal agencies, and to review those with an understanding that they are going to approve technologies that make technologies, rather than approve one drug, one bug assets. This is a huge shift in the governance and the way that the FDA is managed. And we should anticipate some really dramatic changes in both the speed and the alacrity with which these packages are reviewed. Great question. This is happening as we're speaking here today, so it's pretty exciting.
That's great, Michael. Thank you. Mark, I think the next one, we'll start with you. Do these rapid vaccine development platforms only offer options for stopping influenza and/or SARS-CoVs, or can these platforms meet needs for other pandemic and potentially emerging infectious disease?
So strictly speaking, yes, the whole objective of rapid vaccine development platforms is that they should be target agnostic. So they should be flexible, so you could actually use them against different types of diseases. And actually at VIC currently, we have the same platform being used to develop a vaccine against Q fever, which is a disease particularly prevalent amongst our forces, our military forces, and in specific countries, which is actually a bacterial infection. The platform that we're developing is broadly applicable to viruses and bacteria from that point of view, and viruses beyond COVID-19 and influenza. And in fact, in our first 120-day demonstration, we went after the virus called Lassa fever virus.
I think just one point to add to what Mark was just saying is that different platforms, different methods of approaching this problem will have inherent disadvantages or advantages, strengths. And so making a somewhat diversified investment into having more than one dominant platform available is going to be absolutely essential. The correct answer to the COVID problem will not necessarily be the correct answer to the influenza or the Coxiella or the Lassa fever or the Zika problem. And so having a monolithic investment is inherently hazardous for a large number of reasons.
Thank you, Patrick. Also, Patrick, another question that might be in your area of expertise for sure. There's a question about variants and mutations in the spike protein that help them evade the antibodies generated against the spike protein, either through natural infection or a vaccine. The issue there, I think, is twofold. I think what the question is asking is, if it's getting around the spike protein, what are other approaches? And I think it's broader targeting, but you're certainly the expert on that. As they get around the antibodies produced by either natural infection or vaccination. Natural infection is different, of course, because it's broader than just the vaccine. But maybe you can touch on that point real quickly, Patrick.
And I would ask Michael to chime in as well and add his points. But I think it's important, one, to understand that the current vaccines that are out there, as Michael said at the very beginning, is one vaccine made 8 different ways. They're all delivering essentially the same educational material. It's the spike protein. And so for those vaccinated individuals, the immune response that they mount focuses the selective pressure, that is the selection for variants, on the spike protein. And it gives the virus a number of escape hatches. And what's important about our platform and other rapid response platforms, that they are intentionally designed to not focus on only one set of epitopes, one protein. You want to prevent having to play this game of whack-a-mole. You want to close in the evolutionary opportunities for the virus by not affording it the opportunity to mutate only one protein again and again. And the other thing that you see, as you were pointing out, is in the course of a natural infection, yes, you experience the breadth of all of the different antigens that the virus has, because the virus is much more than just the spike protein. Your immune system learns to recognize all of the other proteins that the virus has, and it recognizes the infected cells that way. But inherently, over time, your response to the virus through natural infection will wane. And what we know from MERS and from SARS is that period of time that you're protected post-infection is directly proportional to the severity of the disease that you experienced. So if you had a mild case, chances are, at least from SARS and MERS, you are much more susceptible to reinfection in a shorter time frame. I don't know if Michael or Mark, you want to add to that
You said it beautifully. The only thing that we would recommend now is multiple groups, including the VIC, are anticipating the escape mutants of the future and building in advance of their occurrence vaccines that will counter them. So we're not only in a reactive posture. The way in which we work globally with our partners, including difficult partners in China and Russia, still have an open door that inform us about the evolution that they're seeing, so that here in CONUS, we can design countermeasures, vaccine targets that will counter the variants which haven't even reached American soil. This is a huge phase change in our capability and is a perfect blend of structural biology, obviously vaccine diplomacy internationally, and the capabilities of multidisciplined biology physics teams like the VIC and others. So great point.
Thank you. I know we're starting to bump up against it, but there's a few more questions here that I think would be helpful to the group. How is the rollout of testing for variants versus the original COVID strain been? How can this be improved, as many testing centers are still only testing for the original strain?
Yeah, so I'll just take that to begin with, just to say that in addition to the virus working out its way around our immune system, it's also ironically working its way around our testing systems. The testing systems that are dependent on the sequence being specifically correct. If that sequence changes, then you can start to miss variants. And that has been identified as a problem. Now, what has been addressed again for the first time, and the resources being put behind that, is obviously sequencing nationally, sequencing intensively to find out what are the variants that are out there that are dominating, and how do we have to redesign our PCR-based tests and potentially other antigen-based tests to be able to still capture the positive infected individual with testing. And it is, again, one of these things, just like with having a nimble vaccine program, you do actually have to have a nimble testing program as well that reflects what the sequences are that are out there that you actually need to test for. Michael, you may know even more about this area. I find it amazing what the virus is capable of doing with regards to evading our testing.
Yes, it'll soon be reported that we have 2 strains that defeat our testing. The point is made here for those of you who have responsibilities for bioterrorism appropriations, is that there are technologies that have been demonstrated where the bad guys actually take out the part of the genome that our PCR tests use. They're called detector defeat biological munitions. Now, this has happened in the natural case where the viruses that are more difficult to test continue in the community and propagate. And we have this occurring in Hungary, France, and Brazil at this moment. So we do have viruses that are actually not just evading vaccine immunity, but are actually evading the very tests that are the cornerstone of our public health response. There's a separate group of 10,000 people and 10 companies working on these new diagnostic technologies to detect any pathogen, either that from nature or at the hand of man, which has been engineered to evade detection. This is a hot area, and the rest of the discussion can happen in a skiff.
Thank you. Next question is, can you please explain what cytokine storm is and how it can be minimized?
Michael put a quick chat into the just in text form. Cytokine storm, if we were to simplify it, it's an exuberant or overreactive response, usually to a pathogen the patient has never seen before. And what it is it's the fire of the immune system, the acceleration of the immune system that does not stop. It's not just a little fire, it's a fire with gasoline put on it. But unfortunately, unlike in the normal case, we don't have the brakes to slow it down. The reason why your fever goes away is because your body has suppressing or dampening capabilities. So cytokine storm is a break in that beautiful link between inflammation and immunity to control that infection, and then knowing when to stop and to bring the soldiers back home. So this is an uncontrolled immune response that really needs therapeutic braking or well-designed vaccines that produce judicious and balanced immune responses rather than over-aggressive ones. Great question.
Yeah, that's and a great answer. Thank you, Michael. Actually, the questions have been terrific, and there's a couple more here that I think are also very pertinent to what we're talking about. Since the vaccine is still considered in the experimental stage, what, if any, is the acceptable level of risk to the population as it goes into humans when it's no longer experimental?
So I think the point is in the slide that showed the preclinical and clinical path from hamster studies to non-human primate, if necessary, as called by the FDA, and then into phase 1, 2, and 3 studies in humans, which we hope to commence in the second half of this year, basically. And obviously, the acceptable level of risk is that it has to pass through the FDA's regulatory path here to emerge safe and efficacious after phase 3 trials so that it could be submitted for an EUA. Again, at that point, I guess after phase 3, in a sense, it's no longer experimental in a formal sense, but we'll still be learning about it as we do learn about the current vaccines, even under the EUA.
Thank you, Mark. And one thing that's worth pointing out is as you developed, your team developed this platform, your eye was really to safety first, which is what to the delivery system versus the adjuvants that Patrick was talking about, for example. So it was designed safety first, something that the immune system wouldn't react negatively to, and in fact would enhance the immune response. So it's a great question, but it's biology and it's hard to predict what'll happen, but the whole platform was designed, in this instance at least, with safety in mind. Another question here, which I think is terrific: how would this technology platform contribute to the prospects for a combined universal COVID flu vaccine with durable immunity? Companies are already thinking about this long term, and you may get better compliance. So, Mark and Michael, I don't maybe tackle that if you would, please.
Maybe Patrick, it's another very, very good question. Patrick, do you want to take this?
I mean, I'll start, and I can see Michael nodding, so I know he has a point. But I think this is a very important bridge, right? This concept of having a joint influenza COVID vaccine to improve compliance. And I have several close colleagues that are working in this arena, and it's exciting what they're working on. I think, though, it's a bridge, and it unfortunately admits defeat to the virus in the sense that our objective is not to have a continuous vaccine program where every 6 months, 12 months, 24 months, you have to roll out a new vaccine. And that platform presupposes that you're going to have to persist with annual vaccinations for both flu and COVID-19. I think the goal here is to be able to deliver that, to deliver tailored epitopes as needed. But as Michael was alluding to earlier, to stop the evolutionary opportunities, to really limit the chances for the virus to continue to persist, so that you obviate the need for that joint vaccine platform altogether. And the goal of a universal flu vaccine, that is to say a flu vaccine that you have to take once every 5 years, 10 years, is also a very much ongoing arena of work that some of those COVID hybrid platforms are leveraging right now. But I think that our goal is a little more ambitious than just constantly having to update every 18 months or 12 months what the vaccine platform would have to be. I'll let Michael and Mark chime in.
Very, Patrick, beautifully stated. The thing I would add is that when you're developing vaccines, your perception as a clinical operative is that the whole entire population of the US are your patients. So you cannot do a lot of horse trading and allow one population to suffer more than the other. To this end, the decisive knockout that wipes out COVID-19, 2021 and beyond is the super play. So how do you do that? Well, you do that through international collaborations to look for those rare winners that are impossible to infect with COVID. Why use just mice and just other animals who are giving you the solution of the mouse and the other animal? We need the human, what we call the clinical truth. And in the current processes that Vic and others are involved in, we're finding those rare, uninfectable people. We're in the laboratory, we cannot infect their cells with the new variants. And then we are reverse engineering their natural solution. And by doing so, Patrick Reeves and the rest of the Vic team and companies like this are producing super play vaccines that go after the current threat, but also include a target that is found on all coronaviruses, therefore producing a secondary backup immune response to protect against variants. That's the decisive knockout. For those of you on the Hill who are looking for these investments, they're under the prophecy appropriation for preventing pathogen evolution. So those are the ones that are being earmarked under the current administration as a priority. So look for them. They are where our innovation is timed right for the biggest moves on our chessboard against COVID.
Thank you, Michael. And we're down to our final question, which I guess is really the question of the hour. Do people who have been infected and now have natural immunity need to get the vaccine? And if so, why?
I'm compelled to take this one on because my patients that were vaccinated with foreign vaccines, these are US operatives operating in Asia, were infected quite heavily in January, February, March of last year and had really impressive immunity. Many of these were airline crews flying into China. They would get reinfected in the US all through the winter. So their natural immunity, which tends to be very broad, that means very good, unfortunately slowly decreases over time. We cannot give you the number of survivors of COVID infection 1 that get reinfected from COVID from a second strain, months later, but we will tell you that the variants have a higher success rate in breaching those people that were naturally immune. And that's what worries us.
That's a great answer. Thank you, Michael. And I think that is it for the Q&A. So thank you everybody for your astute questions, and thank you Doctors Callahan, Posnanski, and Reeves for your participation and your insights, and to the G2G folks for getting this all set up. So if there's additional questions, can always follow up with the G2G folks, and it'll get to us. And thank you everybody for making the time today.
Great. Thank you, everybody.
Bye-bye. Thank you.